up those having Wi-Fi problems? We're good? Mostly okay, good. And we're getting lots of questions through, and also comments. Okay. Wi-Fi is a popular question. Okay, so we'll get cracking now. So, firstly, I'd like to welcome you all here today. It's a real pleasure having you here to come and join the Christmas lectures, which, when we first ran these in 2019, we were hoping were going to be an annual thing. And obviously the COVID pandemic hit. So this is the first one we've run since our inaugural one, so this is the second one. But it's great having you all here, and we'd like to give you a very, very warm and wintry welcome. And just before we really begin, the Slido thing is addressed. We're all happy with Slido. Anyone not happy with Slido? Good. Okay, the second thing I want to say is that in case of emergency, we're not expecting any test alarms today, so if the alarm does go off, we'd like to sort of evacuate the building, and the fire exits in this room are the two doors back there, and there's a door also here for those in the lower tier and on this side of the room. So when if the alarm does go off, please follow the fire marshals, which will be wearing, who will be wearing big bright yellow vests, and they will direct you out of the building at the nearest fire exit, and then the convening point is about 100 yards towards Russell Square, outside the old nurses' hospital. Okay, so without further ado, I would like now to welcome our co-director of education here at the UCL Great Ormond Street Institute for Child Health to give you a very, very warm welcome, and that's Jane Simmonds, Professor Jane Simmonds, everybody.
This one here. Well, welcome everyone. So this is what it's like to be at university. I don't know whether this is your first time, but it's such a fantastic time in your life. So all of you are clearly thinking about this, aspiring to coming to university, and I'd have to say that this university is one of the best in the world. So we hope that you choose UCL if we're providing the type of thing you're interested in. And the fact that you've come today to learn about the research that we've got on offer means that you may very well be thinking about coming to UCL. So we have got, well, thanks to Daniel and the team for putting on this event. I don't know how much you read about it in the program, but this institute is one of the biggest centres in the world for child-related research. So we have a very significant number of researchers in this department who are influencing medicine and primary science across the world. We also provide education as well. So we run mainly postgraduate courses here, but alongside our colleagues in global health who run the undergraduate programs in this building, particularly, we provide that. So I'm just going to talk a little bit about what you've got on offer. I hope you've enjoyed looking at the posters. I know I saw you all talking to the various researchers, and I hope you found those interesting. And now we're going to launch into our Christmas series. We've got 3 of our researchers who are going to be presenting aspects of their research. We've got one of our postdoc researchers, this is Annie Etuk, who's been researching in the area of blood-related malignancy. We've got Maria Caluianu, who has been exploring rare kidney diseases, so we're going to hear about that. And then we've got a very topical area from one of our associate professors who's been researching here and teaching here for many years, talking about the microbiome and its relationship with mental health. So I'm going to hand back now to Dan to welcome our speakers. Thank you very much.
So thank you very much, Jane. Okay, so now I'll introduce myself. So I'm Daniel Grayson. I'm a postdoctoral research fellow here at UCL, Great Ormond Street Institute of Child Health, and I investigate Duchenne muscular dystrophy, which is a devastating muscular disease. But I'm also the lead of the public engagement focus group, and in that capacity, I'm welcoming you all here today. And the first speaker that I would like to introduce to you today is Annie Etuk. So Annie has spent 6 wonderful years, as you may have read in the programme, developing gene-edited cell therapies for the treatment of childhood malignancies, and she states here that she's been very, very happy to work with a number of colleagues, and that's one of the most amazing things, always working with people that you enjoy spending time with. And she's going to come here and talk to you about her research today, and I hope that you find it fascinating and inspiring, and it helps you make a decision about your potential future, to potentially do science rather than medicine, as I saw one particular question earlier. So I would like to welcome Annie to the stage now.
Sometimes it takes a little bit of a nudge. Just trying to...
Minor AV issues. Do bear with us. It worked earlier, obviously.
Yeah, it's just a bit dusty. Let's try it. Yeah.
BJ? No, it's just HDMI. No.
Yeah, it's coming up. Okay.
Okay, Annie Etuk, everybody.
I think somebody asked... I think somebody asked the question, Imperial or UCL. The answer is UCL, 100%. 100%. So it's really nice to physically see you in person again. You all look beautiful, and I'm absolutely honoured to be giving this lecture today. As Daniel said, I'm very lucky to work as a research fellow in Professor Kasim's group here at the Institute of Child Health, and our research does focus primarily on producing cell-based therapies for the treatment of leukemias. So I'll be giving you a crash course to CRISPR and gene editing. So by the end of this lecture, we will have covered the basics of DNA, genes, and mutation. There'll be an overview of what CRISPR is, and we'll discuss how this system has been used to edit genes in the lab, followed by a brief introduction to our group's research and how we use gene editing tools like CRISPR to create universal T cells. And there'll be some discussion on the ethical considerations of gene editing. So, the cells in your body contain a complete set of genetic material that you have inherited from your parents, which we refer to as your genome. And now the genome is squashed into little structures called chromosomes that you may often see mapped and arranged like this. Now, chromosomes consist of DNA, which is made up of building blocks that we call the DNA bases. Now, you can think of the DNA in your genome as an instruction manual that makes you who you are. Your DNA is split into sections that we call genes, and these genes code for characteristics such as your height or the texture of your hair. Now, genes are also translated into proteins that can influence how certain cells develop and how they function. Now, sometimes there are mistakes in this instruction manual, mutations in gene DNA that can potentially lead to disease. And these mutations could be inherited, spontaneous, or caused by a mutating agent such as UV. Now, the mutation may translate to an altered form of a protein that no longer works properly. And if this protein is essential for cell development, then the cell may not survive. Or alternatively, the cell could proliferate out of control, like we see in cancer. And scientists can determine what mutations cause disease and what changes to make to ameliorate these conditions. And that's where CRISPR comes in. So the CRISPR-Cas system is a tool that scientists can use to change or edit DNA within a genome. It was discovered in bacteria and is part of an inbuilt immune system that protects bacteria from invading viral DNA. So this bacterial system was adapted for use in the lab by a number of scientists, including recent Nobel Prize winners and all-round science queens, Jennifer Doudna and Emmanuelle Charpentier. Now, the system we used in the lab comprises two components. A small molecule of RNA that acts as a guidance system and is therefore referred to as guide RNA. We like to think of guide RNA as a sat-nav homing in on a specific DNA postcode. The second component is an enzyme called Cas9 that can cut strands of DNA like a pair of molecular scissors. So together, these two components form the CRISPR-Cas complex. Now, when the CRISPR guide RNA finds its DNA target, it locks onto that specific section and it slices both strands of DNA. And this cut can then be exploited in order to edit what was originally there and therefore correct a mutation. Now, there are gene editing platforms that predate CRISPR-Cas, such as TALENs and zinc finger nucleases. They are also two-component systems. However, in these earlier editing platforms, the guiding and cutting components are fused together, making them complex and expensive to synthesize and requiring a new fusion protein for each mutation that you want to target. So, dare I say it, the sexiest thing about CRISPR-Cas is that the guiding and cutting elements are separate, meaning that you only have to customize simple pieces of RNA to guide you to the mutation and then provide this common Cas9 enzyme to cut at the targeted location. So CRISPR-Cas is faster, easier, and cheaper than what came before. So targeting and cutting with CRISPR-Cas means that you can introduce a break in DNA at a specific location and exploit this break to insert, delete, or modify genetic sequences. And that is the fundamental principle of gene editing, a DNA break followed by some form of correction. Now, when you introduce a break in DNA, the cell will try to repair the damage, and there are two mechanisms or pathways that can be triggered when DNA is broken. So in the first pathway, the DNA is cut, the gene is broken, and it doesn't stick back together properly, so it essentially disables the gene. A broken gene means a broken protein, so we can use this pathway to prevent protein production, for example, or to turn off or knock out a receptor. Conversely, in the second pathway, Cas9 makes the cut, and we, at the same time, provide a new piece of DNA to patch and repair the gene sequence. This way, we can introduce or knock in a precise gene modification. So furthermore, the CRISPR-Cas system has now evolved to allow what we call base editing. So with a slight modification to the Cas enzyme, we can now precisely swap out a specific DNA base. And in this example, we have swapped a DNA base from a C to a T, much like the find and replace function that you might use in a Microsoft Word document. Now, on to our specific research. So for the treatment of leukemia, we can take T cells and engineer them to express a chimeric antigen receptor, or CAR, for example, CAR19. Now, CAR19 targets a protein on tumour cells called CD19, and when engaged, our CAR T cell will kill the tumour cell. So in clinical terms, we can take cells from a patient, modify them to express CAR19 that targets their cancer, and then reinfuse them back into the same patient. So when we use patient's own cells to make these CAR T cells, we refer to this as an autologous cell therapy. We don't have to worry about finding a match. We don't have to worry that the cells will be rejected because the cells came from that patient in the first place. But using a cancer patient's own cells to make CAR T cells can sometimes be difficult because patient cells can be fragile, they may not expand well, and we are limited with the amount of starting material we can take. Now, what CRISPR and gene editing has allowed us to do is take cells from an unmatched, healthy donor, modify them to express the CAR and target their cancer, and then gene edit these cells to knock out receptors that would otherwise recognize the patient as foreign. We thereby prevent rejection of unmatched cells. So removing the potential for rejection means you can use any old donor and give these cells to any old recipient without having to worry about finding a match. That's why we call it a universal therapy, and that single product can be used for multiple patients. Now, a word of caution, because CRISPR and gene editing is a relatively new and powerful technology, and anyone who's ever seen a Marvel film knows that with great power comes great responsibility. So I have so far described what we call somatic therapies, where we edit specific cells, like a T cell. The edit is contained within the T cell. No other cells are affected by these edits, and these edits will not be inherited by children or subsequent generations. So somatic cell therapies have been researched, tested, and regulated for several years. In contrast, editing germline cells like sperm or embryo is relatively new. Genetic modifications made to embryos can be inherited, which therefore raises some safety and ethical considerations about what we edit and why. So gene editing of embryos leading to a birth is prohibited around the world. However, in 2018, Professor He Jiankui from China announced the birth of gene-edited twins. He had used CRISPR on the embryos to knock out a protein that's involved in HIV infection. Now, this announcement quickly made mainstream news and was met with widespread condemnation because, well, first of all, it was illegal. Second, he wasn't transparent about his procedures or investigating potential risks. And probably most importantly, other medical interventions are available for the treatment of HIV that do not involve tampering with the germline. So Professor He and his mates, they went to jail. And I think it's really important that we, as genome engineers, talk about the controversial stuff because it's these sorts of things that will make headlines. We need to be transparent in order to highlight all the good that gene editing has achieved, including proper world firsts, like saving this little girl's life. Her name's Layla. She was treated back in 2015 with gene-edited T cells here at Gosh. And this was the first time in the world that gene-edited cells had been used in this way. And hopefully there are more lives still to be saved, as we currently have 2 clinical trials open. One for the treatment of B-cell leukemia, like what Layla had, and our newest trial, which opened a few months ago, another world first. It uses base-edited cells for the treatment of T-cell leukemia. And I'm incredibly proud that I was able to contribute to opening these trials. So there you have it. In summary, we have covered the basics of DNA, genes and mutation. We know a little bit more about CRISPR and how this system was adapted for use in the lab, including to generate these universal T cells. We briefly touched on the ethical considerations of our research when we discussed that naughty rogue, Professor He. But overall, we discussed the overwhelming good and showed how gene editing really, really can save lives. So, thank you very much. I absolutely have to acknowledge Professor Kasim, who started it all. Our group lead, Christos Georgiadis, he's also another good egg. And all of my wonderful, wonderful colleagues in Team Wasim. And of course, there are a variety of funders who make our research possible. And indeed you, thank you very much for listening. And I look... Thank you.
Amazing. So well done.
Shall I pop it back to...
Fantastic. Incredible. You did an amazing job. What do we all think of that then? Did we like that? Am I not speaking loud enough? We all like that one, right? Okay, now we're going to hear from our second speaker, who is Maria Caluianu, and she is going to talk to you about rare kidney disease. She's a PhD student. She's in the second year, just in her second year of her PhD, and she's already achieved lots of amazing things, including she is the UCL-wide champion of the 3-minute thesis. And what that essentially means is that she is amazing at explaining scientific concepts in a way that people understand and be accessible to all types of audiences. So it means that you're in for a real treat today. And so, without any further ado, I introduce Maria Caluianu. Thank you, Maria.
Thank you. All right. So, hi, I'm Maria Caluianu. I am a PhD student, just finished up my first year of my PhD, and I'm going to tell you a little bit about my project. Now, first things first, my supervisor told me that I don't have to make this a particularly Christmassy talk, just because it's called a Christmas lecture. So the first thing I did was make it as Christmassy as I possibly could. So let's proceed scientifically. I've identified the top 2 components of a good Christmas story, okay? Firstly, there has to be an underdog, according to me. And secondly, there has to be a group of friends that go on to help that underdog. So those are the 2 things you should be looking out for, because that's what I'm going for, okay? Now, what is my talk about? That's my PhD title. It's a big, scary word in the middle of it, which you can just choose to completely ignore, because what I do is quite simple. I look at a rare kidney disease, I try to understand it, and then I try to treat it. And that rare kidney disease is called arthrogryposis renal dysfunction cholestasis syndrome. But you can just call it ARC syndrome, okay? So ARC syndrome is a rare and really severe disorder, actually. So babies born with ARC syndrome have problems with their joints, their kidneys, and their livers. They can also sometimes have problems with their skin, and they can have bleeding problems if they get a cut. They just keep bleeding and they don't clot properly. They can have problems with their hearts and also with their brains. And unfortunately, before they reach the age of 1, they tend to pass away. So this is kind of as severe as a disorder can get. So what causes all of this? Well, actually, all of this is caused by one single typo in one single gene in a child's genetic code. So how does one single typo do all of this? Well, to understand this, you have to understand that in every single cell in your body, you have a system that's a bit like train tracks that gets molecules to where they need to go. This is called the endolysosomal system. Think of it this way, okay? Let's say a cell is London, and each one of us is a molecule, okay? The endolysosomal system would be the London tube. So people have jobs, right? The tube gets them to where they need to go. They do whatever they need to do, and they keep London running. Now, what this one little typo that I was talking about does is it takes out a really important bit of the tube tracks. So all these different trains start to derail, yeah? So it's Monday morning rush hour, and half of us are stuck underground in the dark, and the other half are waiting at a platform for a train that's never going to arrive. London grinds to a halt. What does this mean in terms of cells? Simple. The molecules don't get to where they need to go in the cells, so they can't do their jobs there, and so the cells don't work. And because all your cells in your body, from those in your bones to those in your kidneys, have this system, all these different types of cells don't work. Which really explains the symptoms of the babies I talked about before, right? And why they have problems in all these different organs throughout their bodies. Now, what do? I look particularly at the kidney, and I look at where and when in development these trains start to derail. And then I look at the consequences of the crashes. And later on, what I'm going to do is I'm going to use a special treatment, a bit like what Annie was talking about, to fix that typo I talked about in the genes of these children, to fix the kidney. Now, that's all good. We've understood that. But in order to really understand my project, you have to understand the kidney, our underdog of the day. This is the organ that keeps all the good stuff in your body while getting rid of all the stuff you don't want in the form of pee. Now, why is it our underdog of the day? Well, the kidney is often a misunderstood and kind of neglected organ. Yeah? It's not as cool as the brain or as lovable as the heart. Yeah? And a lot of people think of it as some boring old filter that just makes pee. So not as many people study it. Now, over the last year, I've heard my colleagues try to make the kidney sound more engaging by using different metaphors. So I've heard it's a sieve, it's a coffee machine, it's plumbing, and my absolute favorite, it's an octopus. But it's none of these things. It's lace. Its beauty lies in its intricacy, and you can really only appreciate it if you look at the details. So let's look at the details. This is a nephron, the basic functional unit of the kidney, right? You get 1 million of these, put them together, and you end up with a kidney. These things are about 3 to 5 centimeters in size, okay? So we're already seeing some of the complexity that I was just talking about. Now let's look at how these nephrons work and think about how they might stop working in the disease I study with all those derailed trains. Okay, so first things first. Blood gets to the nephron right there, yeah? And here, there are some cells that I do have to admit look a little bit like octopuses, okay? So they have these long tentacles that are entangled together and held together by a special glue. And these tentacles, held together by the glue, stop big molecules like proteins from leaking from your blood into your urine, okay? So what happens when all those trains derail in these cells, the octopus cells? Well, maybe the glue doesn't get to the legs of the cells. Maybe the legs start to unravel. And maybe big molecules like proteins start leaking out into the pee. Okay, let's go a little bit forward to this part of the nephron. This is the proximal tubule. At this point, small molecules that get through that octopus sieve thing that I just talked to you about get sucked back up into your blood. How does that work? Works a little bit like this, okay? So let's say this side, in yellow, is the inside of your tubule, yeah? The inside of the proximal tubule. And this side, in pink, is your blood, yeah? And these two purple dudes are transporter proteins, okay? And let's say the small molecule here in the little blue boxes is a sugar. Now, this guy on this side, his job is to always take sugar and take it from outside the cell and put it into the cell, yeah? The other guy, his job is to take that sugar from inside the cell and put it outside of the cell. And when they're standing like this, everything's going great. All that sugar is sucked back up into your blood. But what happens when the trains derail? What if these two guys swap positions? Or what if they're left stranded wandering around in the cell? Mayhem. So all of the sugar that's meant to be powering your body starts leaking out into your urine. That's no good. Now, in the disease I study, both of those things happen. So we have big molecules like proteins leaking out into your urine, and you have small molecules like sugars or amino acids, which are the building blocks of proteins, also leaking out into your urine. Now, when I was told I would be one of the only people in the world studying this disease, and I would probably be the only person looking at the kidney, I panicked a little bit. But then I started the only way I could think of, which was to look at kidney cells in a dish and to try to mimic the disease in these cells. Yeah? But there's only so much you can do with cells in a dish. There's only so much you can see, right? This is basically string. I need lace. I need cells to behave like they do at home in the kidney, naturally. But it's really hard for me to get samples of the actual kidney. Yeah? I mean, this is a rare disease to start with. And then secondly, these kids, they have problems with bleeding. So taking a big needle and taking a sample out of their kidney, it's really not an option. So what do? I need a kidney, but I don't have a kidney. Simple. I build a kidney. So that's what I started trying to do, sort of. I started trying to build a proximal tubule, the part that sucks up all the small molecules like sugars. Okay, so how did I do that? First, you have to understand that I see the proximal tubule as essentially two tubes that sit next to each other. The proximal tubule tube, that has all like the sugars in it, and the other tube, which is the blood vessel tube, into which the sugars go. So I needed to make a device that looks a little bit like this, that helps me make two tubes that sit next to each other in a jar. And then after I designed it, I used a machine a little bit like this. That's a 3D printer that prints in 3D. So it prints layers on layers on layers. So it makes a three-dimensional object. And I made this device that lets me make those two tubes next to each other. Now, the next step for me is to add cells into that. So I want to put proximal tubular cells on one side into one of the tubes and blood vessel lining cells into the other tube. Okay? And then, hopefully, the cells will behave much more like they do at home in the kidney. And I can mimic the disease in this particular model. Maybe I can even test out some potential treatments in this model. Speaking of potential treatments, the next step will be to use a special treatment to fix that typo in the kidney's genes and to make sure that the kidney is happy and functioning again. Now, as you've been listening to me, you might have noticed that it looks like I'm looking at a really narrow part of science, yeah? I'm looking at a rare disease. In that rare disease that affects all these different organs, I'm looking just at the kidney. Inside the kidney, which is affected in many different parts, I'm looking mainly at the proximal tubule. And so what I'm doing can seem kind of like insignificant and small. That is until you take a step back and you look at the big picture. Because science and progress don't happen in isolation. They happen when groups of people, all working together on their small pieces of the puzzle, come together to make a bigger solution. And so I'm going to use my lab to illustrate that as an example, and how they can take my project from this smallish thing to a much greater thing, yeah? So let's start off with Seth. He works on using ultrasound, the things you use to look at babies, to instead look at the kidney. That is a kidney. It looks pretty cool. And he looks at this so that he can use this to be able to target the kidney and inject treatments, for example, my treatment, directly into the kidney. Now, if he's successful, this is the difference between me getting my treatment just to the kidney or me getting the treatment all over the body, which can have a bunch of side effects that you don't want. So, best of luck to him. And speaking of treatments, he, alongside other colleagues, including Will Mason and Claudio Cosmescu, work on other treatments that help fix genetic typos. Now, let's say my treatment doesn't work out. I can work with them to maybe make a treatment that does work to make these kidneys function again. And as for my engineered piece of lace, my little proximal tubule that's in 3D, well, the sky's the limit in terms of expansion. In our lab, we have everyone from people who work on making really realistic versions of blood vessels to people who work on making that octopus filter part that I told you about earlier and making that in a dish. Now I can just physically connect all of those together at some point to make an even more representative model of the kidney and test out treatments and mimic diseases in them. So there you have it, the final component of the Christmassy story, okay? The group who comes together to help the underdog, because that's what science is. It's groups of people coming together to change the face of medicine, one organ at a time. And thank you so much for listening. This is the rest of my lab.
Maria, that's wonderful. Thank you so much for that talk, Maria. So, are we all feeling inspired? Yes. Sorry, I can't hear you. Are we feeling inspired yet? Yes, we are. Excellent. Do you want to feel even more inspired? Yes, of course you do. Let's feel more inspired. Okay, so to inspire you further, I would like to invite another one of our amazing colleagues onto the stage. I would like to invite Mona Bajaj-Elliott onto the stage. Mona. Mona is an associate professor here at Great Ormond Street Institute of Child Health, and she really wants to understand how microbes that live within our bodies influence our health. And she's very good at this, and she's going to tell you a really nice story about why this is important. And we know that she's really good at it because she sits on an expert panel for gut microbiota and health at the British Society of Gastroenterology. So we know that she's good.
Oh, thank
We're
you so
really
much.
looking forward to her amazing talk. So take it away, Mona.
Well, I hope I, reach your expectations. Thank you, everyone, for hanging on. I must say before I start, the best comment you're going to hear all day came from Annie. What future career you choose, make sure you don't go to jail. I think that's the one we should remember. Okay, so you've heard two stunning talks on personalized medicine, whether it's a rare kidney disease or CAR T-cell or lymphoma, all this turning into universal therapy. I want to swing completely the other way and go to population health. Our Institute of Great Ormond Street Institute of Child Health belongs to the Faculty of Population Health, and UCL actually, because over the pandemic, was actually right at the front of mental health investigations. So these are some of the headlines given to me by the, the publicity department of UCL, the media. And I'll just like to go through some of them. I'm going to start with the one on the Telegraph and move clockwise. So grandparents' mental health suffered during lockdown. Study shows they were forced, those who did not meet the grandchildren, showed more depressive symptoms. I think that's absolutely what one would expect. And the one on Mail Online, it says UCL experts examined over 30,000 new parents here. And I think what was interesting, the comment here was that 300 couples were stuck with postnatal depression simultaneously. And I thought that was quite important because everybody concentrates on the mum all the time, but here's the dad or the other partner also having issues with depression. So I think these kind of studies do support and try to think of what's the way forward in terms of carrying on understanding mental health and improving outcome in people. Here's BBC, depression, at Christmas, if you're not meeting your loved ones and you've been in lockdown. So the overall message was that we've all had a psychological hit because of lockdown and anxiety, job losses, you name it. It's I think, affected all of us to a certain degree. I want to finish this slide by showing the top. So what has been recommended, again, I guess from UCL studies, yoga should be prescribed for older COVID sufferers. Mindfulness, physical and creative activities. I guess going out to a park and having a nice walk is much better than being cooped inside. And of course, if you look on this side, which I did not know, the young people who prescribe surfing and dancing by NHS. Your young people, has any of you been involved in surfing and dancing? Anyway, so, which is all good, it's all good. However, I just want to, in my next slide, move on to a different topic about mental health, because you want to come from all directions to improve your health. So, the topic that I've chosen is, because I work on microbes and mental health. Is there a link? So, as Dan said, I'm Mona Bajaj-Elliott, and I've been here for quite a few years, and I'm really interested in gut health. So you might think, why is a gut scientist interested in microbes and mental health? So I hope I can convince you in the next few minutes, 10, 15 minutes that I have, that actually it's the gut that is the link. And I'm very, very passionate about it. And I hope that you also will leave this room thinking, there's a little bit more to my gut and what I eat that might be having an effect on my overall health. Okay. I have a couple of slides which are, I think we have to go through them because we all know about microbes now. And all we see in popular press, or they only come into conversation, is when they've done something bad and ugly and caused disease, I guess. And we have to remember infectious diseases still kill more than 10 million kids around the world every year. So I'm not denying that we don't have a big problem with infections. We do have. Okay, but that's one side of the coin. Just a few terms. An endemic infection is like malaria in sub-Saharan Africa. It's there. You know, we can't get rid of it. We have outbreaks. You heard about the migrant who sadly lost his life with diphtheria in Kent in the last fortnight. We have a streptococcus A infection outburst going on. The 9th child died yesterday. Incredibly sad. So, you know, these infections keep re-emerging. So, you know, we all have to keep a consistent eye on microbes. You can have an epidemic, which involves a few countries. An example of that I've given here is the Middle Eastern Respiratory Syndrome, which was the precursor to SARS that we had in 2020. This was in Saudi Arabia in 2012, and it was a jump from camel to humans, but it was very well contained, and it only was, well, they did very well. And it spread as far as the Middle East and India, but it did not take over the planet. Unlike pandemics do, that's the terminology of pandemic. And the most famous is, of course, in the 14th century, the bubonic plague and Black Death that wiped out 50% of the European population. And it's called Black Death because that's what it does to your organs, peripheral organs. And it's called bubonic plague because that's the swellings of your lymph nodes. So we have to keep infection away and the bad guys. Yes? So fast forward from 14th century to 2022. I mean, I must admit, that looked really nasty. If SARS had looked like that, I think we might have all run off to have our vaccine ASAP. So let's keep that right, yeah? Okay, so 2022 is nearly November, December now, the 7th. So we're still facing infectious challenges. You know, the SARS-CoV-2, the Omicron at the top left, has evolved. We are approaching winter with flu pandemic. You know, who knows what's going to happen? And the NHS is already in a crisis, as you know. There's still lockdowns going on in China until last week. I'm sure you've been hearing that in the news. And now what we are left with is this challenge of lingering mysteries of COVID. We know about long COVID. Nearly 1 million people in the UK are suffering long COVID. You know, we have to understand what microbes do to our bodies. We know that viruses like SARS do something to our brain. That's called brain fog. 1 million people being unwell is not a pleasant thing. That's the challenge for you future scientists who want to stay in science. You know, that we have to solve these issues. This year a new virus reappeared, monkeypox, which has got a new name called mpox since last week. And you can see that gentleman's face with poxes on his chin. And it's an infection you can catch if you're close to some people. But we were very fortunate because we still have the vaccine for smallpox, which works for mpox. So it's absolutely fantastic. So my favorite word on this slide is vaccines. It's the only medication you have it before you get any disease. Isn't that incredible? And we live in a rich country enough to have vaccine available on a shelf. How fortunate are we? But in spite of that, vaccine uptake is a big issue. And again, UCL really is at the forefront on behavioral science. And that's Susan Michi, one of the professors in one of the other departments. Yeah, so all I'm saying is vaccines are the most wonderful drug ever made, and yet we have anti-vaccination going on. So you might have a role in, you know, being influencing on that. You know, there are plenty of job opportunities. Depends what your interest is. Okay, and the last slide on the doom and gloom is we're getting globalization. We travel within hours between London and Australia. We are having climate change issues. And the big issue of climate change is going to be that infections that have stayed in hot countries like Zika, Ebola, are going to start moving up into the northern hemisphere because we are getting warmer and warmer. That'll bring different challenges in the future. And the last one here is antimicrobial resistance. So we have this strep A outbreak going on, and this morning in the news, the government is talking about is there enough penicillin to give to school children so it doesn't become a big problem. Too much giving of antibiotics is raising antibiotic resistance, and we will not be able to treat infections. So the question is that a good idea to give antibiotics to all kids in a school? And Dame Sally Davis, who was actually one of the AMR leaders, has always said, predicted, by 2050, if you don't sort that out, you might have as many as 15 million deaths. We did have 6 million for COVID, which is still 6 million too many. Okay, so that's the background, knowing and appreciating and respecting microbes, that they are there to cause trouble as well. But surprisingly, majority of them are actually good. You know, you could name 50 infections, 100 maybe, and I'm going to show you how many trillions of bacteria live in our gut. So 100 infections over centuries of evolution just shows actually it's quite hard to become a bad guy. It's the good guys that rule. Okay, so good guys, just going back, food industry. All this baking we do, we use microbes, right? Whether it's in food, cheese making, wine, beer. So these are all microbes at the end of the day. What will we be without them? So we're using them from our cookery skills, and yet at the same time, we've got everything in fridge freezers to make sure the bad guys don't multiply and cause trouble. How much do fridge freezers, how much is that contributing to climate change? Just to keep these guys away. The other good thing microbes have done is it exploded this new field of molecular biology. You know, everything you've heard, and I will come back to Maria and Annie, you know, this CRISPR-Cas and diagnostics, it's all based on bacterial and viral biology. So we are actually exploiting them in some ways for our own betterment. So I think that should be acknowledged for the whole planet. You know, we're making GM crops. How are we doing that? What tools are we using? They all come from microbes. Okay. We all know about PCR now, don't we? Where did the enzyme for PCR come from? It's from a bacteria that lives in hot water, the Taq polymerase. Yeah. And now we're using microbes to do other wonderful things for us, to actually digest waste, digest plastic, any oil spills in oceans. We want to use microbes to be able to, you know, eat them up. So I just wanted to again highlight that there has to be proper respect for microbes. Okay, let's go to the link that I talked about with mental health. So it's actually a lot of, you know, very mathematically inclined people have worked out that here's the sky with very little light pollution. They say there are about 10 to the 11 stars in our galaxy. And there are at least 10 to the 12 bacteria in our gut. Okay? And that 1 trillion is 1,000 billion. There are only 8 billion people on this planet. So you've got nearly 100 times more bacteria in your gut than there are humans on this planet. So that is extraordinary. And sorry, Maria, I don't think pee is that interesting as much as poo is the best, okay? Okay. The other thing we've learned, of course, is it's surprising how much of your biology is going on in your guts, guys, okay? And if only we paid more attention to it, we'll all be healthier. But the point I'm trying to make here is that if you count the number of bacteria and the amount of genetic information they're carrying and the chemicals they produce, compared to that little human there, which only expresses 20 or 25,000 genes. So when I do my research, I don't think, oh, I've got all these bugs in my gut. I think, no, I'm living amongst these microbes because I'm much more smaller. Okay? Those bacteria have enough capacity, metabolic capacity, as the liver has. So we're all carrying an extra organ that we never actually spend time thinking about. Okay? Right. So this is a slide produced by one of my students, Nicole, who was actually working in the lab during the pandemic. So she gave a talk for an antibiotic resistance festival. So this is part of that slide. So just very simply, we are child health. We look at, you know, early life. So a happy gut. The main take-home messages, depending on what you eat, why should not, why do bacteria have to go searching for food when they can just go directly in you? Because the food is there. It's as simple as that. So a happy gut, it depends on what you're eating, I guess, quite a bit of it. And the lesson is what you want is high diversity of bacteria, different types of bugs, which she likened it to be like a nice, lush garden, okay? A happy garden. And on the other hand, if you look at the poor child who's got a sad gut because of disease like cancer, we heard about cancer already, radiotherapy, chemotherapy is killing all the good things in you as well. Antibiotics. Antibiotics are a killing machine. They don't know whether you're good or bad, so they kill everything. So you don't really want to be on antibiotics because work from UCL has shown that if you take one course of antibiotics as adults, your gut bugs never come back for up to a year. So you're making yourself susceptible to other things. So a bad, sad gut, only the bad guys will grow then, right? So look at the red section. It's got low diversity, means very few, and you can equate that to a desert. Nothing grows there. Something will come up, a few cacti, for example. And that is not what you want. So our message is, to take your gut from a lower diversity to high, and we want to change a desert into more of a lush garden. And I think that really needs to be brought to the public as well, rather than always worrying about, too much hand washing. What are you doing? You're removing all the nice antibiotics your body makes from your palms. You never get an infection on your palms. Why? It's as simple as that, because our body makes lovely, all that sweat that comes off. You actually make your own antibiotics. So there's a balance. It's an ecosystem. Okay, gut-brain axis. So I guess it's very simple. If you have a happy gut, there's a big nerve called the vagus nerve that connects to the brain. And all the chemicals being produced are, of course, going to affect brain health. It's not rocket science. The only problem right now is we still don't understand what those chemicals are. So a lot of more work to be done, and maybe that's where you might be interested in sorting out what those trillion bacteria are doing in the gut. Okay, so I just want to focus on one pathway, and that's tryptophan. Tryptophan is an amino acid, which I'm sure you've all learned all your amino acids, at the school. So there are different types of bacteria. There are 3 types here, which actually digest this tryptophan and make different chemicals. And one of them is serotonin, the happy hormone, we call it, right? Which is actually quite reduced. And if you don't have too much of it, it leads to depression. So, tryptophan. Okay, and then it makes this one I wanted to highlight, IPA on the left. And if you look at its properties, it's a neuroprotectant. It activates your brain cells, glial cells, astrocytes. It's an antioxidant. And I was really pleased to read that it also inhibits, the last line, inhibits beta amyloid fibril from formation. So I guess maybe you already heard Alzheimer's disease is plaque formation. And this good guy actually reduces that. So there's another connection with another disease of the goodies that these microbes make. Okay. So I'm showing data from my own group on the right-hand side. And these are healthy lab adults. We all did a quick experiment from the stool sample, I'm afraid. And what we did is got a profile of these different tryptophan metabolites. So the pink is the tryptophan. You can see everybody makes it in different amounts. The yellow is serotonin. Okay? And person number 7 there has got no serotonin. But I was quite pleased to see they make a lot of IPA, the blue ones. At least they're making this guy. So even diagnostically, if you could start looking at these metabolites, just get a picture. They're walking around healthy. I mean, they are PhD students. I bet they're under stress all the time. So a bit more, and these are sort of reasonably healthy. When we go into patients at Great Ormond Street, and we have done very limited work, it doesn't matter what you're looking at, whether it's a cancer patient, intensive care, undergoing transplant. You only see tryptophan in their guts. Everything else just crashes out. It's the first thing that goes. And what does that mean? That means all the bacteria that help to metabolize and break down tryptophan are gone. They're just not there because of your treatment. So how bad is that for your health? And that's what we want to change. Okay, so because it was gut, mental, brain axis, I just wanted, yesterday I did a quick PubMed search, and there are at least nearly 49,000 papers out there. So it's a vast area to cover. But so this topic was really to give you a flavor of how much we need to understand better. So with lots of various titles, I thought I'll just give your attention to the last one. Association between mental and oral health in Spain, a cross-section study, more than 23,000 people aged 15 and over. So I thought because of your age group, I must highlight that. So we all brush our teeth at least once, twice a day. What are we brushing away? Microbes. Yeah? People who have bleeding gums, we already know that bacteria from your mouth can go into your bloodstream and cause heart issues. So, why? So that's not surprising. If you drink alcohol or smoke or take drugs, immediate effect is from your mouth to your brain. So why should not all the microbial chemicals not affect your mental health? It's not a surprise. We just don't know what they are right now. So again, it's more stuff to be understood. Okay, I'll finish off on therapeutics because that's at the end of the day, we all want to be healthier with our mental health and try to understand this axis. Experiments are still done in mice because we can't do them in humans. And if you could find the right bad guys, then you can come in with a phage that can target a virus, that can target and kill the wrong bacteria out of your gut microbiome. You don't want to disturb all trillion bacteria. You just want the bad guy out. That's affecting mental health. You could improve your health by your nutrition, prebiotics. I'm sure you all know about prebiotics and heard probiotics in the news. These are actual microbes. People, what is bioactive yogurt? You all have yogurts, yeah? Everything is bio nowadays. What is it? It'd be interesting to hear if somebody knows what those microbes are in those yogurts, for example. You can buy symbiotics, which is giving the food and the bug together. And there is a new place called psychobiotics. It's a whole new billion-dollar industry. Again, a lot of young people are setting up companies, in the microbiome world. Go and check it out on Google. And maybe that's the way you might want to go forward. And here is, at the end, I like to use the word, so this therapy is called fecal microbial transplant. And that's actually poo transplant from one person to another, right? You might go, ooh, yuck, okay. But we also have, nobody's going to say no to a blood transfusion. So what's wrong with a fecal transfusion if it makes you well? Why not? By the way, the first fecal transplant happened in New York nearly 65 years ago. There were 3 patients who were at deathbed with an infection from C. difficile, and the doctors had given up. There were no antibiotics that were working. They came in with a donor fecal transplant. It's an enema in endoscopy, and within a week, all 3 went home. Okay? Now, the reason it didn't take off is because for pharma, there's not much money in that. But it's come back in fashion, and our colleagues at King's College who study end-of-life liver disease are doing fecal transplant. It's become routine in America and the UK. And guess what? People who have undergone fecal transplant, their depression just disappears. Again, pointing out that the microbes have a major effect. Okay, so it is a billion-dollar industry for lots and lots of conditions: autism, schizophrenia, epilepsy, you name it. Everybody is working on it. So to finish off, I like to say the gut microbes are not just linked to your mental health, but to your physical health as well, okay? They are central to your life. It's the most intimate relationship you'll have in your life, not with your partner, because they live with you, okay? So when you next time put a burger in your mouth or a donut, remember, that's the microbes are going to have that as well. So feed yourself well. And I guess, have you heard about personalized immunology? I think we're going to have personalized nutrition as time goes on. Get your own personalized microbiome. I think that's the best way forward. Okay, and then that's exactly what's happening. If you read papers on mental health during COVID, time for personalized nutrition based on your gut microbes, okay? So before I finish off, this is only a 20-year-old area of research. It's up and coming, but it's the tip of the iceberg, and there's so much we do not know, which is below the water. And maybe you as future scientists might be interested in unraveling a lot more of these mechanisms and improving human and plant and animal health on this planet. Thank you.
Well, that was a wonderful talk, I have to say. I thoroughly enjoyed it. And I'm feeling intellectually enriched today after those lectures. Are you feeling intellectually enriched? Yeah? Yeah? Are you feeling nourished with knowledge? Are you feeling nourished with knowledge? Excellent. I'm glad to hear it. If you can workshop some better alliterative phrases than that, I'm interested to hear them. Who played the hand grip strength test game? Go on, hands up. It can't have only been. There must have been more of you. Must have been more of you. Anyway, we have a couple of winners, and you can win T-shirts. So those that didn't do the hand grip strength test, you're now regretting it. So I'd like to announce the first winner. The first winner is, and I hope I'm not embarrassing myself with the pronunciation of this. It's Kahindi Adamola. Where are you? Where are you? Okay, so the second one is Rayan. Is that Rayan Juma? Well done, Ram. Well done, Hindi. Excellent. So whilst we wait for it, you've all been answering your questions on Slido, right? Okay, so now you've all been asking your questions on Slido. We're now going to have a panel discussion to answer some of those, if not all of those questions, if we have time. So we're going to bring all of the amazing researchers back onto the stage, and they're going to sit with me, and we're going to have a discussion. Okay, let's... Oh yeah, before we... Just a quick reminder, who's been doing the crossword? Have you been doing the crossword? Have you been... Yeah, so when you finish the crossword, when you leave, hand your completed crossword with completed feedback on the back into our delightful colleagues as you're leaving via the entrance at the top, and they'll give you a prize. So that's an incentive, isn't it? So just bear that in mind. And also, at the end, we'll all shout out the extracted word. Not yet. Okay. So the first question that I think has been upvoted is quite a relevant question for our talk, anyway. Is medicine worth all the effort? And how would it impact your mental health? And I think I'm going to ask Mona to start with this question.
Okay, I'd rather break that. Okay, I'd rather break that down into two. The first is medicine worth the effort?
All the effort.
All the effort. When I read that first, I was just thinking, if I get on an aeroplane, I've given my life in the hand of the pilot. I like them to be properly trained to make me arrive safely. So I think it is. You, at the end of the day, are the life of somebody is in your hands, and that is a big, big responsibility. I never did want to do medicine because I get too emotionally attached to a patient and feel, it's not for me. So I chose science. So the second part is about mental health.
Yeah. How it impacts your mental health?
By doing medicine.
By doing medicine.
I guess, okay, what we do know is people on night shift have a poorer microbiome because of the internal clock. We all have a circadian clock, and our microbiome also has got a circadian clock. And so there's no surprise that people on night shift have an issue. But if you're aware of how to maybe, improve your microbiome in those years of life, but consultants don't do night shifts, do they? So, you do well at the end of the day. But any job is, what's the difference in doing a night shift, doing medicine, or being a train driver, or a nurse, or, this is new life. So I think whatever you do, you've got to enjoy it and make sure it's right for you, and mental health will follow, I think.
So, Maria, do you have any insights maybe you have doing a PhD impacts mental health?
Oh, okay. It's a lot of work, but I think if you have a nice team, at the end of the day, I think everything's quite okay. I don't know. What do you think?
It's definitely hard, but nothing that's worth doing ever comes easy. I think that if it's something that you want, then of course it's worth it. But it is hard. What I would say is don't be afraid to step away, to take a break, to fail, to try again, to come back. Even if it's something that you really, really want, don't be afraid to step away and say, this isn't working for me right now, but to come back to it.
So since you've got the mic there, I'm going to ask you the next question because it's relevant to your talk, which is, can gene editing increase my height?
That's a great question, and the short answer is no. Sorry about that. It's a good question. I mean, if something like height, something like kind of those physical characteristics, they are multifactorial. So it's not one gene that you can potentially fix that will give you what you want. I mean, if that was possible, I would CRISPR my hair straight, I'd CRISPR my fat cells away, I'd CRISPR my genome and replace it with Beyonce's. Do you know what I mean? It's no. Sorry, the answer's no. The answer's no. Sorry. Did anybody want to add to that?
I guess if no one else wants to answer that, then I think another very relevant science question is for Mona. So, Mona, how would you perform a fecal transplant in the most sterile way? Are there not harmful microbes which can transfer over to the patient? Really good question.
Yes, thank you very much who raised that question, and it's an important one. Nice to meet you. No, absolutely. At the moment for fecal transplant, they do screen for all known pathogens. Okay, that's is done. Absolutely. Now, if you are at the 65, 70 plus kind of age group, and if it's going to save your life, the big question is, what about the pathogens or would-be pathogens that we don't know are already there? Right? It's the silent ones. So at that age group, I think, if it was my parents, you say, well, what the hell? I've only got X years to live. I'll just get on with this therapy. And people do that for C. diff, for example. Now, the question is, and this is something we do talk about a lot, being at a child health hospital, that if you could have a fecal transplant, you have to give to your child, and there might be many unknowns, and this person has got the future X decades to live. There's a big, big question mark. Absolutely. But what we are trying to promote for babies and children is that when a child is diagnosed with cancer, for example, we know they're going to have chemotherapy or radiotherapy, for the parents to really go out of their way to improve their microbiome. Okay? And then they go in for their treatment, and their microbiome is demolished because they're on antibiotics. It's completely killed off. But we have already stored a sample of their microbiome. And to speed their recovery out of hospital, then to give them their own autologous microbiome back. But the slight challenge, it's a big challenge. It's taken 5 years to get permission by the regulatory bodies, nearly 5 years for adults. So doing it for children, most probably I'll retire before that. But that's the intention. And no parent would refuse the child having their own stool sample, or of a brother or sister. Keep it in the family. But yes, great question. Thank you.
So actually, the next one is for Maria.
Okay.
Okay, so Maria, what motivated you to investigate ARC syndrome in the livers of babies? Kidneys of babies, I think. I often conflate liver and kidney.
No. So actually, I didn't choose the topic. The topic kind of chose me. I said I wanted to do gene therapy, and this was the disease that was kind of suggested to me to do gene therapy on. So that's how I ended up with ARC syndrome as my PhD topic. Yeah.
Amazing, and I think that's a really salient point to make. Actually, it's something that speaks to my own experience as well. So, I kind of ended up having my subject find me based on my other experiences around becoming a scientist. And I think you might find the same if you go study science at whatever level you do. You'll end up having a similar kind of experience. You might become inspired by a lecturer or something else supervisor. Okay, so Annie, are there any limitations to cell-based therapies at the moment? And what is being done to overcome them? If you want to have a think on that.
Yeah, Mona made a very good point. It is quite expensive. It's a very expensive process. I think one of the things that we are doing to overcome them is investigating things like universal therapies, because the same amount of effort and time that you put into creating a bespoke therapy for one patient, you can use the same amount of time and essentially the same amount of money to make that a product that can then be used for multiple patients. So that is maybe something that we are trying to use to overcome those expenses.
And just following on from that, there's another really interesting question here. Do you think gene editing of babies will become legal in the future?
That is a really good question. I don't know what the world is going to look like in 50 years' time, but I hope that there is never a scenario where babies will be edited for kind of cosmetic purposes. For health reasons, perhaps we will move towards a future where that is more commonly done. But for cosmetic purposes, I can't see that happening, and I think I wouldn't like to see that at all.
Thank you for that answer to that question. So there's another one here that I think is for you, Maria. How long will it take to develop a treatment plan for Ormond disease, and will you be able to benefit those suffering the disease today?
So to the last part of the question, okay, so it will take a long, long time, and it will not help those babies that are currently suffering from ARC syndrome, most likely just because the lifespan of the disease is quite short, and it takes years and years and years to do clinical trials, to do everything. And we have a disease that's really complicated, that affects lots of organs, and all those organs need targeting, not just the kidneys. So, yeah.
Fantastic. Thank you, Maria. Just one more sciencey question, and then we'll just change tack a little bit and answer some more personal questions. So, Mona, why do you think psychobiotics is getting so much popularity?
The answer that comes immediately to my mind is we have become a pop-up pill generation, right? We want immediate, let's go to the pharmacy, give me this, and I'm better. And people have found ways of making good money out of it, and why not? We have a young new company set up from 3 UCL students, by the way, all female-led, who are harnessing breast milk microbiome, and they've set up their own company, Booby Biome. They're based upstairs, and because 1 in 10 child is born preterm, and there are lots of mums who can't breastfeed their babies. So there is a demand, and once you see that demand, you go for it. So yes, there are lots of good things. When psychobiotics, mental health is such a big thing that if it must have an impact on economy. So, let's get everybody feeling better and making, helping towards the growth of the country. So I think I can see why it has got so much attraction.
Yes, yeah. So there are these drugs, nootropics, right? They are kind of these drugs which help us concentrate and focus. They've become huge industry.
Yeah. How much coffee can you drink, right?
How much Ritalin can you take, right? Okay, so the person that's asked me if we can talk about my research, you can come and talk to me at the end of the night, explain something, but that's not the main focus here, even though I would be happy to talk about it. But we'll change tack now, and we'll just ask some more questions about maybe the career path that our esteemed speakers have taken at this point in their careers. So, beginning with a very simple question, what were your undergraduate degrees? Beginning with whoever would like to answer first.
Biochemistry.
Biochemistry, here we have one biochemistry.
And the reason I fell in love with biochemistry, I'll share with you, it was during my A-levels, and it was a biology lesson, I think, and it said glucose uptake, glucose gets phosphorylated, and guess what? It can't go out again because it's lost its affinity for its transporters. And I thought, how beautiful to explain something that simple that we take for granted, but chemically explain that. And that was it. I had to do biochemistry after. Because at the end of the day, no offense, genes are important, but the real activity is the proteins. So there you go.
She's wrong. She's wrong. The most important thing is physiology, which is bringing everything together.
I know. Okay. 1-0, you've been there.
Maria.
All right. So I did applied medical sciences, which is biomed with a twist, I guess. So that was me. Yeah.
I did medical biochemistry, which is biochemistry with a twist. I also was very lucky to do an industrial placement year as part of that degree. And I would absolutely, I think somebody asked about work experience. I would absolutely recommend grabbing both hands any work experience opportunities that you can get. I did the same for my undergrad degree.
So I think we have a work experience opportunity here at ICH. I think maybe some people attending today did that work experience. And it's great to see them back here, if indeed they are here. I saw their names written down on the registration sheet. So we do a week-long work experience, which I think, Mona, you were involved in. So if any of you are still haven't quite left your school and have gone up to university already, then it's something to consider for next year. We do a work experience scheme. So that's the kind of work experience that I would recommend. And also I think that you probably agree with me, wouldn't you?
Actually, can I just add to that? The slide I showed you by Nicole, she was a UCL student, spent the summer with me. Then she got an internship at GSK for a year. And now she is going to Harvard to do immunology. So there are lots of opportunities, you know, and yeah, so take them with both hands. Any opportunity that comes along.
So there's perhaps more kind of philosophical questions that I'd like to pose to one or all of you, which is, why are doctors so underpaid when they play such a significant role in people's lives?
Gosh, that's not
Do
a
you have
question
any
for me.
opinion on that? I think it's maybe even a political question.
I guess you're talking about junior doctors. Because consultants are not badly paid.
Consultants are not badly paid. So if you reach the top, you're not badly paid.
I think my slight issue with this kind of comment is that because they are so significant for society, what about everybody else? You know, everybody is doing an important role. So I hesitate in that term. Yeah, but take up medicine, it'll be less to begin with. And, you know, I think surgeons are multimillionaires. So, you know, it depends where you want to go. If money is the driving force, take it in that direction. Set up your own company. With a medical degree, you'll have more, more investors will be interested in you.
Well, I think there is just such a range of pay scales depending on where you are in your career, how far you go, how successful you become. It's very difficult to really answer such a question as that. And the only thing I don't know if you would agree, the only thing I would say is when you're deciding what career you want to go into, I would try and find something that I'm really, really passionate about. And just really use that as the barometer for making your decision on what you do in the future. I mean, I did that and I don't really have any regrets. No. I'm not multi-millionaire and I'm quite happy.
But your passion has sustained you, and that's
my passion sustained me. Yeah. For artists, for example, it might not be the case. So yeah, good question. Yeah, you can ask that, Mona.
Yeah, I was just going to say, you'll spend more working hours at work than you will with your partners at home. So it's a relationship you have with your work life. So, like any relationship, it breaks down, you separate, choose a new direction. But, but passion is the main thing. And without that, it doesn't matter how much you're getting paid, it won't be fun. Do you want to have fun in your working life? Of course.
Okay. So, I think we're really, really sort of starting to run out of time now. So I'd just like to sort of maybe have some closing comments from the panelists. Do you have any advice just generally that you would say to someone starting their career in science?
Yeah. Well, I'm the oldest of you lot, so I'll tell. When I did my degree and I was your age, we just fumbled around. I'm absolutely amazed now looking decades forward how much mentorship goes on, how all your social media, your news is at your disposal. So really, the decision then relies completely on you, and and I think you are in a great place. Unlike if I go back to the 80s, You just did what you felt passionate about, and that was it. There was no other choice. So you've got a lot more choice, guys, and make the most of it.
Advice. I don't know. Do what makes you happy. Don't do something that makes you miserable. You're not gonna be happy, really. Like, imagine staying like 12 hours in a lab on a project that you hate. You're gonna be unhappy doing that 3 years. So don't do that. Don't do things because you're gonna get a title, because it's prestigious. Do things because it makes you happy. So yeah.
Yeah, I absolutely echo that sentiment. Do what you love. Don't be afraid to step away when you stop loving it, but also don't allow your insecurities to prevent you from grasping every opportunity. That's it.
Yeah. You want to say something? No.
Would you like to add?
No, yeah, I would echo all of those sentiments, and the things that you mentioned earlier about doing what you're passionate about is what I would say. Yeah, I would just echo those points. Okay, so I think all of our panellists have done a wonderful job today, and I think they all deserve a really warm round of applause. I'd like you all to give yourselves a round of applause for being a wonderful audience, for getting amazing questions from the side. Made it a really vibrant session. I thoroughly enjoyed it. I thoroughly enjoyed having you. The only thing I'd say, there's two things I want to say. First is, do stay in touch. If you've got any questions, then feel free just to pose those to anyone working here. We've got this whole session has been filmed, so if there's something you've forgotten partway through that you want to refer back to, then you'll be able to find this on our YouTube channel, which is on YouTube, UCL Child Health.
Can I say something before you close? Hi guys, one last comment. Can you join me to actually thank Daniel for all the hard work here as well? Thank you, Daniel.
And one final thing. Before, I know you're desperate to stand up now because you have been very patient sitting down for a long time. Please hand in your feedback forms for a prize. And also, just did anyone get the extracted word? G. Well done, everyone. Thanks very much. Oh, yeah, it's great. I was just a bit... The AP system here was shit. And I'm just worried that the reason a lot of them were sort of breaking down into private conversations is because they weren't hearing it as well. Obviously, it was often when you were talking about things like poo, but that was great. I forgot, but I meant to do a straw pop.